Good afternoon, children. In last class, we discussed mutations. In this mutation, chromosomal structural mutation, numerical mutation. Numerical mutation, what we discussed here, aneuploidy. Here there is a mutations, what we discussed, chromosomal mutations. Chromosomal mutations. In these chromosomal mutations, what we have discussed here, chromosomal structural mutations, and the chromosomal numerical mutations. Chromosomal structural and numerical mutation. Structural mutation, there are 4 types we discussed. Last class, what is the mutations here? Deletions, duplications, inversions, translocations. These 4 in last class we discussed. Chromosomal mutation, structural mutation, deletion, duplication, here inversions, translocations. Last class we finished. Now numerical mutation. Chromosomal numerical mutations. Here, what is the numerical mutations? There is 2 types. One is aneuploidy, another one is the euploidy. Euploidy. Numerical mutations, aneuploidy, euploidy, 2 types are there. Then here, aneuploidy means, what is the aneuploidy? The addition of chromosomes to any one set of the cell. 23 sets are there. Out of these 23 sets here, any one set is added. Then that can be called aneuploidy. Addition of single chromosome to any one set of this here cell, that is called as this here aneuploidy. But in this addition or minus subtraction, if we add, that is called hyperploidy. If we remove, that is called as the hypoploidy. Hyper means adding chromosome. Hypo means removing chromosome. Here we last class what we have discussed, hyperploidy, trisomics, heterosomics. Trisomics example, Klinefelter syndrome and Down syndrome we discussed in aneuploidy section. And next one, what is there here? There is a hypoploidy. Hypoploidy example, what is hypoploidy? This aneuploidy, hyperploidy is there and hypoploidy is there. Hyper means 2n plus 1 or 2n plus 2, the adding chromosome. Hypo means here reducing 2n minus 1 or 2n minus 1 minus 1. So here if from the original set of chromosome, one chromosome is removed, that is called as the monosomy. The formula of monosomy 2n minus 1. Removal of total set, complete set, 2 chromosomes are lost, complete set is there, nullisomics. Normally nullisomics cannot survive. Monosomics only can survive. What is the monosomic, nullisomic? Now we can discuss, followed by euploidy. You can see here, this is a cell. Hyperploidy, last class we discussed. Hypoploidy. Hypoploidy means, you can see, this is a cell. How many sets will be there? I told 23 sets, like this first set, second set, third set, fourth set, fifth set, sixth set, seventh set of chromosome, 8th set, 9th set, 10th set, 11th set, 12th set, 13th set, 14th set, 15th set, 16th set, 17th set, 18th one, 19th set, 28th set, 21st, 22nd. This is 23rd set. From this 23rd set, there is a first 2 here. What is this here? This is the 28th set, 21st, 22nd, 23 sets are there. From this set, what is the provision? Here, 1 may be removed. Actually, from each set, 1 may be removed or 2 may be removed. Maximum capacity, what is this each set? From this set, what is the maximum capacity? 2 can be lost. First choice, what is this here? 1 may be lost. Suppose from this 1 is lost, that is written as 2n minus 1. Complete set is lost. Here 1 chromosome is lost, 2n minus 1. Complete set is lost. It can be written as 2n minus 1 minus 1. 2 times we can write. Non-formally, it can be written as a 2n minus 2. Suppose here from any one set, one chromosome is lost, it will be called here monosomics. This can be called as the monosomics. And from this any one set, complete set is lost. Suppose this set or this set or any set can be lost, 2n minus 2, or it can be written as 2n minus 1 minus 1. This is how it will be called here. This is called as a nullisomic. It is a nullisomics. So monosomics and nullisomics. Normally, monosomics there is a choice to survive. Here they can survive. Monosomics can survive, but nullisomics cannot survive. Why nullisomics cannot survive? Suppose you see here you want the genes one set you observe A, B, B, C, C. Each one 2 copies are there. Every gene 2 copies. Suppose here this gene 2 copies are lost means those characters can be lost. Therefore, those characters cannot produce in our body means it will not survive. Suppose one copy lost, one more copy is there. So that to perform activity, this copy can do. Therefore, no problem. And fourth here, one chromosome is lost from set, no problem. But complete set is lost, organism cannot survive. Therefore, here, nullisomics cannot survive. But monosomics can survive. Monosomics formula 2n minus 1. So example for monosomics you see, that is the here Turner syndrome. Turner syndrome is the example. What is the Turner syndrome you see here? This is a female cell. Mutations here, monosomic. Monosomic formula 2n minus 1. 2n equal to 46. Here in man, in human being cell, in man cell, what is this here human being cell? Human being cell 2n equal to 46. N equal to 23 sets. So here how many sets are there in value? 23 sets. Each set 2 chromosomes are there. So here it is a set 2 chromosomes, set 2 chromosomes like that. 23 sets are there. So 2n equal to 46. 2n minus 1 means here 45. So what chromosome can be lost here? In female, human female you can see. This is a human female, it can be seen. In human female, what is the 23rd set? X chromosome, X chromosome. What is the 23rd set here? 2 X chromosomes are there. But one X chromosome is lost. Then it can be represented as a 0. 0 means there is no chromosome. How it will be written here? These all body chromosomes can be written as autosomes. Here it is a body chromosome. These are all chromosomal sets. How they can be called here? Autosomal sets. The last one, how it will be called? Sex chromosomes. In human female, what are the sex chromosomes? X and X. But here, due to the monosomics, one X is lost. Therefore, it will be called X0. What is the karyotype of monosomics here? A plus X0. That means one X chromosome is lost. Out of 2 X chromosome, one X chromosome is lost. That can be called monosomic. It is called as the 2n minus 1. So you have to write what is the karyotype of this here. Monosomic, especially what is the example for monosomic here? This monosomic is called as the Turner syndrome. This is how it will be called here, Turner syndrome. Turner syndrome. Here, last class we discussed about Klinefelter syndrome, Down syndrome. But this class, what is this here? Turner syndrome. What is the Turner syndrome formula? Here, 2n minus 1. The karyotype, it is also written as AA plus X0. The meaning is AA plus X0 means here autosomes all are present. Sex chromosome out of 2 sex chromosome XX, one X chromosome present, one X chromosome absent. It is represented as a X0. Simply here it is in abbreviated. How it will be written? 2n minus 1. So here due to this Turner syndrome, but what is this Turner syndrome occurs in male or female human being. But what are the characteristics occurs due to the Turner syndrome? They have a short stature body, webbed neck, and here they are not able to produce the ova. Infertile female simply. Klinefelter are infertile male, but Turner syndrome are infertile female. Turner syndrome, infertile female, unable to produce ova, so unable to give birth to children. So they are these sterile females, and there is no secondary sexual character. There is no development of breast, poor development of the breast. Pubic hairs cannot be produced. And next one here, hips cannot be developed on attaining the maturity. Like the secondary sexual characters cannot be developed due to the Turner syndrome. What are the Turner syndrome characters here? You see this Turner syndrome. Short stature body, monosomic 2n-1. In this, that is called Turner syndrome. What are the Turner syndrome characters here? Short stature body. Short stature body. The body is a short stature body. And webbed neck. Webbed neck. Shielded chest. Chest is not developed, and this is called shielded chest. Chest is not developed. The breast cannot be developed here. Shielded chest is there. Neck is webbed neck, means very short neck and short stature body. They don't grow more than 4.5 feet. Their height range is 4 to 4.5 feet. Very less height is there. Therefore, short stature body, webbed neck, shielded chest is there. And poor development of ovaries. Ovaries cannot be developed well. So, poor development of ovaries. Poor development of ovaries. Ovaries are not developed well. There is a poor development of ovaries. Ovaries are poor developed, shielded chest, webbed neck, short stature body. Next one here, another case. What would be there here? Sexual hormones. Secondary sexual characters could not be developed well. There is poor development of secondary sexual characters. Poor development of secondary sexual characters. Poor development of secondary sexual characters. The secondary sexual characters are nothing but, according to your visible characters, we can identify whether that person is male or female, or we can identify. That is secondary sexual character. Male secondary sexual character, that shaving, that means beard and mustache hairs. These are the secondary sexual character, pubic hairs. But female, development of breast, development of hips during the maturity period. But on attaining puberty, 12 to 14 years old, that is the puberty. Attaining puberty, what characters are coming? Secondary sexual characters can be developed. So these characters could not be developed in Turner syndrome. Because why? Female sexual hormone is low level. Estrogen hormone low level. Therefore, what is here? There is a low level of female sexual hormone. Low level of female sexual hormone. Next one, another thing here. What is another point? Low level of the female sexual hormone, poor development of secondary sexual character, poor development of ovaries. They are sterile women. Ultimately, what is here? They are the sterile women. They are sterile female. Simply, how they can be called? They are the sterile female. Why this is sterile female? Unable to give birth. Birth could not be given by them. Why? Because here there is no hormones, no development of ovaries, no proper release of ova. Therefore, here there is no regular cyclical changes. Menstrual cycle could not be done regular manner. Therefore, here it is impacted. So here, what is this? Menstrual cycle is impacted. No regular menstrual cycle. No regular reproductive cycle or menstrual cycle. Regular reproductive cycle. These are the characteristics of the Turner syndrome. So here, Turner syndrome, Klinefelter syndrome. Then what are these here? Down syndrome. These are the hyperploidy abnormalities. It is part of this chromosomal structural mutation aneuploidy. Now we are going to euploidy. Generally, euploidy, haploids, no problem. Why haploids here? All sets are disturbed. What is the euploidy? We are going to. This is section covering the aneuploidy. In aneuploidy, hyperploidy and hypoploidy. Hypoploidy, monosomics. -2n-1 hypoploidy. Hyperploidy, 2n+1, 2n+2. Hyperploidy. This is a section relevant with the aneuploidy. Next, we are going to chromosomal structural mutation. What is here? Euploidy. Euploidy. in this case, you can see euploidy. What is the euploidy here? Euploidy is nothing but here there is a numerical change in all set of the chromosomes in the cell, that is called euploidy. Any one set is changed aneuploidy. Euploidy means all sets. Suppose you see one cell. This is a cell. How many chromosomes are there? Suppose 1, 2, 3, 4, 5, and 6. 7 sets are there. This is 2n equal to 14. N equal to 7. 7 sets will be there. One cell. Suppose this is a wheat plant. This is a wheat. In each 2n equal to 14 is there. Normal wheat. This is the normal or wild wheat. Now critical histone is there. There is a exoploidy. 42 chromosomes are there. Wild wheat chromosome 2n equal to 14. Now you can see what is here euploidy. In this, haploids are there, polyploids are there. What is this here? First one, haploids. Haploids means minus. And here polyploids means plus. Polyploids. Polyploid. This is euploid. It is going to euploidy. What is the euploidy? The changes, numerical changes occurs in all set of chromosomes. How many sets are there? First set, second set, third set, fourth set, fifth set, sixth set, seventh set. From the seventh set, you can see here, from first set, one chromosome is lost. Second set, one chromosome is not there. Third set, one chromosome is not there. And fourth set, one chromosome is not there. Fifth set, one chromosome. Sixth set, seventh set. From each set, one chromosome is lost. So here, this is called as here all set. This is going to numerical change. This is going to, all sets are going to changes. Previous class, any one set is going to change, that is aneuploidy. But all sets are going to changes. But one chromosome is lost from one set means remaining all also lost. So the numerical change in all set of chromosome euploidy. But here in these all set here, if chromosome is losing by the loss of one chromosome from all original set of chromosome can form a haploids. Haploids means here 2n equal to 14, here 1n equal to what is here 7. How the haploids are formed here? Total sets are changing means n is changing. 2n will become 1n. Here total set means n is changing. 2n become 1n means haploid. But 2n become 3n, 4n, 5n. There is n plus 1. Single chromosomes are adding aneuploid. Here all sets are changing. Suppose you can see here how it will be written polyploids. Due to the removal of one chromosome from all diploid set of chromosome called as the haploids. Here you see polyploids here. One set is added with chromosome. This is here 3. One chromosome added. First set, one chromosome. Second set, one chromosome. Third set, one chromosome. Fourth set, one chromosome. Fifth set, one chromosome. Sixth set, one chromosome. Seventh set, one chromosome. Each set is added by one chromosome. Then here this 2n will become, actually each set one chromosome is there haploid. Each set two chromosomes are there, 2n are diploid. But each set three chromosomes are there. How it will be called? 2n will become a 3n. And 4n, 5n, 6n and so on. Like this here it is the, what is this polyploidy? Polyploidy means addition of chromosomes to all original set of chromosomes is called as the polyploid. If one chromosomes are added, then diploids will become triploids. Then 2n will become, how this 2n? Modified 3n. 2 chromosome 4n, 3 chromosome 5n, 4 chromosome 6n and so on. So 2n due to the polyploids here, the addition of here chromosomes to their original diploid set of chromosome, these polyploids can be formed. These polyploids 3n, triploid, tetraploid, pentaploid, exoploid, like this and so on, it will be there. So here these polyploids here, again they are divided into two types. Autopolyploids, yellow polyploid. What is the autopolyploid, yellow polyploid? Let us see. Types of polyploids. Autopolyploids and yellow polyploids. Polyploids. These are the polyploids. These polyploids, two types. What is the two types here? Autopolyploids autopolyploids. And yellow polyploids. Autopolyploids and yellow polyploids. Autopolyploids means, suppose here this is a cell, virginal cell. This is the virginal cell, virginal diploid cell, virginal diploid cell, virginal diploid cell. 1, 2, 3, 4, 5, 6. Here this is a 7. But suppose these they can be called as RR. One species, Raphanus species set it in. It can be indicated with how these sets are indicated. RR nature. Suppose here this is RR. What is mean by RR? One R species, Raphanus species. Any species is there, but to represent the genus, I am telling that R. Same species chromosome we are adding. It also R. This also R. Then how it will be called here? Autopolyploid. What is the autopolyploid? The addition of same chromosome to their virginal set, that can be called as the autopolyploid. What is mean by autopolyploid? The addition of same chromosome to their virginal set, those are called as the autopolyploids. Here same RR is there, same we are adding, that how it will be called here? Autopolyploid. This autopolyploid, how it will be called autopolyploid? Autotriploid. These autopolyploids, how they can be called here? They are called as the autotriploids. They are autotriploid. Why autotriploid? Each set 3 chromosomes are there, triploid, so that they are called as the autotriploid. So the addition of here, what is this autopolyploid? The addition of same chromosome to their virginal set. Same means same species. It is the Raphanus sativa. Same Raphanus chromosome only we are adding. Therefore, here same set. So to their virginal set, same nature chromosome added, autotriploids are formed. Here yellow triploids. But what is meant by yellow triploids? We can take this is a, suppose here it is a RR chromosomes. RR, the R species chromosomes. These are R species sets. These are the R species. This set comes under R species. Here. For this RR, what we are adding? Different chromosomes. Suppose B added. Here B added. That means brassica species chromosome. Closely related species. Raphanobrassica. This yellow polyploid is called Raphanobrassica. The addition of different species chromosome to the existing virginal set is called as the yellow polyploid. This is a polyploid, it is a polyploid. Each set 3, but same nature to the what the virginal set is there, same nature chromosome added to virginal set, autopolyploid. A yellow polyploid means what is this? Different chromosome. These are the Raphano means it is brassica, Raphanobrassica. Here you can add what is this? Brassica. Here you can add what is this here? Brassica. Here you can add what is this? Brassica. Here like this here, brassica species. Any other species you can add. What is this here? Yellow polyploid. The addition of same chromosome to their virginal set, then autopolyploid. The addition of different chromosome to their virginal set, yellow polyploid. Like this, what is this here? Polyploids are here autopolyploids, yellow polyploids. Here 2 categories will be there. Like this, aneuploidy, euploidy. Euploids are how many types here? Autopolyploids and yellow polyploids. Total, this is a what is this? Chromosomal mutations. Next one here, gene mutations. Gene mutations here, these are also called as a point mutation. Especially gene mutations, how it will be called here? By this exchange, by the addition or removal of this here nucleotides. One nucleotide is removed or one nucleotide is added. Or many nucleotides are changed. So here we can see this here now, point mutations are next mutation, gene mutations. Another mutations here, gene mutations. Gene mutations here. Gene mutations means these mutations occur at gene level. The mutations are caused due to addition or loss of one or many nucleotides. many nucleotides. One or many nucleotides in DNA are called as, how this is called here, called as the gene mutations. You see this here, gene mutations here. Simply here, any change is there, it is a pen. This is equal to chromosome here, you have to see. Here inside the chromosome, this refill is DNA. Inside the refill in the DNA, this ink is gene or nucleotide, like this organized. Suppose any change happens here. Suppose if you want to break here, like this cap may be damaged. But here there is a severity is there, somewhat intention, refill may be damaged. Ink is much more severe is there, ink may be damaged. Like this, what would be there here? Chromosome level. If it is happens at chromosome level, chromosomal mutation. This here the mutagens are powerful, then chromosome damage followed by DNA. But in the DNA, what would be damaged here? Nucleotides. So this is here, what is this? Nano level it is. DNA nanometers in length, nano level it is. Micrometers in length, chromosome level. So here, where is this here? Somewhat nano level changes are happening. These gene mutations are caused. So you can see this gene mutation where it is there instead of chromosome. These changes where it is occur DNA. But this DNA strand I am taking. The DNA strand, what are present in DNA strand? Nucleotides. The DNA, what is the basic unit of DNA? Nucleotide. How many types of nucleotides are there? 4 types of nucleotide. In shortcut form, we are representing A nucleotide, G nucleotide, T nucleotide, C nucleotide like that. Okay. Adenine nucleotide, guanine nucleotide. A for adenine, G for guanine, T for thymine, C for cytosine. Actually, it is a nitrogen base, but it can form nucleotide. This is a nitrogen base G, form G nucleotide. It is a T nitrogen base forming T nucleotide. C nitrogen base can form C nucleotide. Adenine nucleotide, guanine nucleotide, thymine nucleotide, cytosine nucleotide. Adenine, guanine, thymine, cytosine nucleotides will be there. Now you can see these gene mutations. It is a DNA. This is a DNA. In this DNA, these are nucleotides: adenine, guanine, thymine, cytosine, adenine, guanine, thymine. Here, your wish you can write. These are the nucleotides in the DNA. But what is the gene mutation here? The addition or removal. Addition or removal of one nucleotide or many nucleotides. Suppose here you can see here the addition or removal or replacement. Or one nucleotide is replaced by another nucleotide. Actually, you can see this here GEG. This is a here one codon. Generally, every 3 nucleotides are responsible for what is this one codon. So here in transcription, translation, detailed study is there in molecular basis. But simply you can see what will be there here. There is a here GAG. It will be there. You have to see AGT, CAG, next here CAA, GAG, CTC. Here AGT, CAA, GAG, CTC. Now you have to see by means of mutation. Suppose here you can take a strand. What is the strand here? This is a here 5' to 3', 3' to 5'. It is a DNA strand. What is the complementary nucleotides in this DNA? Suppose here A is there. Here complementary to T. G is there, complementary to C. T is there, A. What is the pairing? A is complementary to T. G is complementary to C. One strand is there. What is the strand you can see here? This is a coding strand here. This is a coding strand. It is a template strand. But here template strand polarity 3 to 5'. This is the template strand. It is a coding strand polarity 5 to 3' here. Now here what is AGT, CAA, GAG, CTC like this is there. But A can complementary to T, G can complementary to C, T can complementary to A, C can complementary to G, A can complementary T, A is complementary T. Again GAG is there, G can complementary to C, A can complementary to T, G can complementary to C, C means G, here D means what will be there A, here G, like this is there. Suppose any change. What is the any change here? Instead of here G, everything is same. Whenever here, suppose you are going to produce RNA, what the sequence is there in coding strand as the sequence will come. But wherever T is possible, instead of T, what will come? Uracil will come. That's only here difference. This is a DNA. DNA during the transcription, what will become forms here? RNA. What is the polarity of RNA? 5 to 3 prime. This is ISD sequence to this here coding strand. This strand is called coding strand. What is the strand? Show the 5 to 3 prime. How it will be called coding strand. What the strand show the polarity 3 to 5 prime. How it will be called template strand. Now you see here complementary sequencing. This is a template strand sequencing, RNA sequencing complementary. Coding strand and RNA equal. Suppose complementary means T means what would be there here? A. C means here G. A means here T, but instead of T, what will come here? U will come. Complementary, A is complementary to T in DNA. But in case of RNA, wherever T is possible, in place of T, what will come? Uracil will come. That's only different. So here you can see A means here U will be there. Next G means what will come? C. And T means what will come? A. T means what will come? A. Next C means what will come here? G will be there. C means what is here? G. And T means what is this here? A will be there. T means what will come? A. And C means what is here? G. G means what would be there? C. A means what would be there here? There is a what is the A? A means here T, but T means U will come. Wherever G is there, C will be there. It is a complementary. Suppose here it is normal process, but one base pair is changed. What is the one base pair? Instead of A, here T will be there. Here one base pair, this one nucleotide is there, A is there, it is a changed. Here next here T is there, but T means here A will be there. Here one base pair, instead of A, here simply only that point only I will give. Here simply this is the DNA. Whatever this here, I am not giving all sequences. Simply here 5 prime to 3 prime, 3 prime to 5 prime here I am giving. It is DNA. Here what is this here? GEG. Here C. G means here. G. GEG codon. GEG codon. Well, this is responsible for right here. GGG is there. This is a GGG, C codon GUG is there. But GUG is responsible for which amino acid? Valine. Whenever valine will come, actually you see, normally this is a sixth codon. First, second, third, fourth, fifth, 5 codons completed. This is a sixth codon. In the sixth codon, whenever normal sequence is GAG. Here normal sequence is, what is the normal sequence? GAG. This is normal phenomenon. You can see normal phenomenon. Normal phenomenon, it is 5 to 3 prime. Here it is 3 to 5 prime. Normal sequence is GAG. But GAG means what is the complementary sequence? C. A means what is here? C. T. Here C is. But what is the codon will come in mRNA? That C means here it is a G. T means here it is a A and here C means G. GAG is there. But JG is there. What will come here in this? Glutamic acid will come. Glutamic acid. Sixth position, glutamic acid. Valine, histidine, leucine, threonine, proline, glutamic acid. 1, 2, 3, 4, 5. Here, sixth position, glutamic acid. This is first position, amino acid, valine, histidine, leucine, threonine, proline. Sixth place, glutamic acid is there. Here, this whenever in the RBC, this DNA relevant is this here, human RBC, these genes responsible for the shape of RBC. Whenever the DNA sequence is GAG, then produce the GAG codon. Then here this GAG codon, how it will be? GUG codon. G means here GTG, GAG, CTC. Now here GAG codon. This is a GAG codon. This GAG codon is responsible for glutamic acid. Whenever glutamic acid is there, the RBC shape is the biconcave shape or it is a spherical shape. Simply what the changes are there, here it is a change. Instead of A, T, what will come? TA will come. In place of A, T has come. In place of T, A has come. Then what would be there here? In place of here GAG, GU will come. Then here this A is replaced by U. One nucleotide is changed. Due to the change of one nucleotide in RNA, instead of glutamic acid, sixth position, what will come? Valine come. When valine come, what is the property can be changed? Sickle shape RBCs turned to the round shape RBCs or biconcave shape RBCs. How it is turned here? There is a sickle shape. That means what is the meaning? Gene mutation. Gene mutation is nothing but here change of one nucleotide base pair or many nucleotide base pair in the DNA can cause these changes. Those changes are called as the gene mutation. Gene mutations takes place at nucleotide level. But in this nucleotide level, one nucleotide base pairs are many nucleotide base pair. You see this one nucleotide base pair, AT pair is replaced by T here. When TA pair is in place of AT pair, TA pair has come instead of GAG, GUG is codon has come instead of biconcave shape or round shape, sickle shape has been formed. So here simple thing, what is the gene mutation? The changes occurs in one nucleotide base pair or many nucleotide base pair. Variations are occur or mutations are occur. That mutations are called as the gene mutations here. But in this gene mutation, by changing of one nucleotide base pair, those are called point mutation. By the addition or deletion of many nucleotide base pair, the DNA structure can be changed. Those are called as the frame shift mutation. Like this here, you can see gene mutation. By changing of gene mutation, change of one or many nucleotide base pairs, change of one or many nucleotide base pairs, one or many nucleotide base pairs can cause mutations. Change of one or many nucleotide base pairs can cause mutations. Those mutations are called as the, how they can be called here? Those mutations are called as, those mutations are called as gene mutations. These gene mutations, they can be called with here different names. What is this here gene mutations here? They may be called as point mutations or frame shift mutation. What is meant by point mutation? They may be called as point mutation, or they may be called as frame shift mutation. Frame shift mutation. What is the here point mutation or frame shift mutation? Point mutation is nothing but here by changing of single nucleotide base pair. One nucleotide base pair is changed. One nucleotide base pair is changed and causes mutation. Such mutations are called as the point mutation. And frame shift mutation. What is the frame shift mutation here? Many nucleotide base pairs. By changing of Here many nucleotide base pairs, 2 or many. Here many nucleotide base pairs. 2 or many nucleotide base pairs, then mutations are caused. Those mutations can be called as the frame shift mutation. The changing of one nucleotide base pair, mutation is caused, that is called point mutation. You can see what is the difference here, point mutation and frame shift mutation. You have to see lines here. Here, many nucleotide base pair. Here, this is a DNA. This is a DNA. Here, A, T, C, A, T, A, T, A, like this is there in this strand. Now, complementary strand. This is complementary strand. A means T, T means A, C means G, T, A, T, A, T is there. Here, one nucleotide base pair is changing. But how it is changing, in place of A, T has come, suppose here. This is, it is replaced with T, one nucleotide base pair. Next here, how we are writing after mutation, T and T, A as it is GT, T is there, T, A. This is, then actually tag, et. But here, tag, triple T, et. Here reading is what would be there, different. Then here, as it is, here 1, 2, 3, 3. So total how many will be there? 8 will be there. Here is also 8 nucleotide base pairs are there. But what will come? Different will come. Instead of here tag, we are reading triple T. It is a tag. Tag is here, triple T is there. This is point mutation. The frame shift means many nucleotides. Here is also maybe added or deleted. Completely, it is in place of one thing. In place of A, T is replacing. This is replacement. But here another case, what would be there? Complete deleted. Here deletion maybe happens here, replacement maybe happens here. So like this, what would be there here? This is a replacement here. But another case, what would be there here? Adding this here many nucleotides. 2 or many nucleotides. Suppose here another case, what is 2 or many nucleotides here? There is an inserted A, T, C inserted. But here, where inserted after A will be there. A as it will be there, but inserted new nucleotide. Here you can see inserted new nucleotide here, TAG and TA. A is there as it. After A, what are adding here? ATC, TAT. Now, how we are reading? Previously tag and here tag. In place of tag, what we are reading? Ta. TAA, ta. The reading pronunciation is changing. By addition of many nucleotides, not replacement. Earlier is the replacement point mutation. One nucleotide removed or one nucleotide replaced. Here it is replacement. First case is the replacement. A is removed, in place of A, T is replaced. That is point. But frame shift mutation. Frame shift mutation means it is not deleting here. Extra or adding or extra removing. 2 or many nucleotides here adding. 2 or many nucleotides, what would be there here? Removing or adding. You have to remove 2 or many nucleotides. You have to add 2 or many nucleotides. Then it will be called frame shift mutation. Point means A is removed, in place of A something T will come. And a replacement is there. No change of frame shift. But here what would be there here? There is a adding many nucleotides or removing many nucleotides. Changing in structure. Suppose you can see TAG. What we are adding? TAG, TA. After TA we are adding ATC. Next, what would be there after ATC? We are adding TAG. Actually, previously it is read as tag, et. Now tag, ta, TCT, et. Like this here, the sequence of nucleotides in the DNA could be changed. So here, what is the gene mutation? By this replacement of 1 or 2 nucleotide base pair, the mutations can be caused gene mutation. Especially point mutation means how it will be caused here. One nucleotide base pair is replaced by the another nucleotide base pair. But frame shift mutation means 2 or many nucleotides added or removed, then the DNA structure can be changed, causing mutation. Those are called as the frame shift mutation. Like this here, frame shift mutation and gene mutation, point mutation comes under the gene mutations category. This is here the gene mutations, the disorders which occurs at chromosomal level. Those are called chromosomal mutations. Here, gene level, those are also called as the Mendelian disorders. These some Mendelian disorders will be there. What are the Mendelian disorders or gene disorders here? Phenylketonuria, sickle cell anemia, thalassemia, alkaptonuria, several gene disorders will be there. And some of those gene disorders, you have a phenylketonuria, thalassemia, sickle cell anemia, three disorders will be there. For you, what would be there? Some gene disorders here or Mendelian disorders here. Thalassemia, phenylketonuria, sickle cell anemia, you have to study in these gene mutations here. And then afterwards, pedigree analysis and X-linked inheritance will be there. Before the type study of this year, thalassemia, sickle cell anemia, we have to discuss about this year X-linked recessive disorders. Then here, X-linked inheritance. Sex-linked inheritance. Sex-linked inheritance. Sex-linked inheritance. What is the sex-linked inheritance? Generally, there is whatever the organism, whatever the cell, there is body chromosomes and sex chromosomes. The genes which are present on body chromosome control body character. The genes which are present on sex chromosome control sexual character normally. But some genes present on sex chromosome control the body character. Those genes inheritance is called sex-linked inheritance. Normal phenomenon, suppose you can see this is a cell. In this cell, these are the chromosomes. First pair, second pair, third pair, fourth pair, fifth pair, sixth pair, like this, seventh pair, eighth pair, ninth pair, 10th pair, 11th pair, 12th pair, and 13th one, 14th one, 15th, 16, 17, 18, 19, 20, 21, 22, 23 pairs are there, like this. But first pair to 22nd pair, first pair to 22nd pair, how it will be called? Autosomes. They can be called autosomes. Last pair, this 23rd pair is there. 23rd pair, how it will be called? Allosomes. Allosomes. This is also called, how it will be called here? Sex chromosomes. Sex chromosomes. Normally, autosomes responsible for body characters. They are responsible for what characters here? Body character. The body characters means here your height is there, your intelligence is there, like this here body characters. Your body character, height is a body character, intelligence is a body character. Then here many your breathing capacity is a body character. So whatever your body characters are there, these body characters are where it is your hearing capacity, body character. Your tasting capacity, this your body character. But vision is there. Vision is also actually body character. But some vision character, vision genes, where they are present? Allosomes. What is it meaning here? Normally, autosomes control body character. Allosomes are sex chromosomes, last pair. What it is controlling? Sexual characters. Sexual traits controlled by the sex chromosomes. But what is the major deal is there? Some genes present on sex chromosome control body character. Here some genes where they are present here, some genes which are situated on sex chromosome control body character. Control body character. Those genes, how they can be called here? Those genes are called as the sex-linked genes. Sex-linked genes. And such genes inheritance is called as the sex-linked inheritance. Some genes situated on sex chromosome but control body character. Those genes are called as the sex-linked genes, and those genes inheritance is called as the sex-linked inheritance. But sex chromosomes, how many types? Two types. X chromosome, Y chromosome. If these body character gene present on X chromosome, X-linked inheritance. Y chromosome, Y-linked inheritance. Both chromosome, XY-linked inheritance. X-linked inheritance, three types. X-linked inheritance, Y-linked inheritance, XY-linked inheritance. You have a study Here, according to your syllabus, what would be there? In sex-linked inheritance, we have a syllabus X-linked inheritance. But in this X-linked inheritance, what is this color blindness? Hemophilia is there. What is the example for this here? X-linked inheritance, color blindness, hemophilia. Let us discuss about this X-linked inheritance. In this X-linked inheritance pattern, you have a syllabus. What is the syllabus? X-linked inheritance. In this X-linked inheritance, what you have, there is a X-linked inheritance you will have. In this X-linked inheritance, what would be there here? There is a examples for X-linked inheritance. Hemophilia is there. What would be there? Hemophilia is there. And color blindness. These two are the examples. Color blindness and hemophilia. Who will be there? Hemophilia, color blindness, examples for this here, X-linked inheritance. Actually, these X-linked inheritance genes present on X chromosome control hemophilia. What is meant by hemophilia? Loss of blood. This hemophilia means loss of blood after injury, during or trauma. When trauma happens, injury happens, loss of blood without clotting. A continuation loss. What is this here? Continuous loss. What is a hemophilia? Generally, if we have any wound is happen here on the body, any wound is happens here, what would be there? Blood can loss. How long it will last here? Maximum 8 minutes. But within these 8 minutes, maximum, some people within 2 minutes, 3 minutes here, it will clot. Those are clotting factors are there. Several clotting factors responsible for this clotting of blood. So here, when any injuries happen, then clotting factors come and sealing the wound and prevent this loss of blood. That is normal phenomenon. But the lack of some genes, or otherwise here the configuration of genes arrangement, what it is doing here, clotting factors cannot produce. Lack of clotting factors, what is the problem? If any wound is happened, blood continuously can loss. So the continuous loss of blood, but what it is doing here, making a disorder called as the hemophilia disorder. What the disorder is caused? Continuous loss of blood here, hemophilia. Here during the injury or trauma, any wound here, continuous loss of blood. But during the injury trauma, continuous loss of blood without clotting, that is called hemophilia. This hemophilia first noticed in Queen Victoria family. In British family, Britain family, Queen Victoria. So that this disease is called as the Royals disease. This which will be called here Royals disease. This disease is called Royals disease. Why? Because here this was first noticed in Queen Victoria family. This is where it is noticed here, Queen Victoria family. Hemophilia disease first noticed in Queen Victoria, therefore it is called as the Royals disease. But here this hemophilia, it is a X-linked. What is the inheritance pattern? We see hemophilia inheritance pattern. Hemophilia inheritance pattern. Suppose you can see this here, male individual. What are the sex chromosomes in human being? Male, X chromosome, Y chromosome. And in the female, what are the sex chromosomes in female? X chromosomes. 2 X's are there. Yeah, 2 X's are there, female, XY is there, male here. So now you can see this male here, X-linked inheritance, male, what are the chromosomes? XY chromosome. Female, what would be there? XX chromosomes are there. Now you can see on XY chromosomes here, what is X-linked inheritance? You can see on XY chromosome male, hemophilia gene. Hemophilia gene, suppose you can see how you can notice here, H for hemophilia. Like this dominant gene is there, they are normal. There is no problem. A recessive gene is there is a hemophilia. The capital letter, what it is doing, dominant allele. And here small letter R for recessive allele. Suppose the gene at dominant condition, hemophilia cannot occur. The gene it occurs at recessive condition, that hemophilia is caused. But here, how many alleles are there in male? Only single allele. This condition of it will be called hemizygous condition. Homozygous, heterozygous. Each gene normally consists of two alleles. Two alleles are identical, homozygous. Two alleles are unidentical, heterozygous. Now you can see this allele is there. This is a dominant allele. Only one dominant allele is there, normal. But this allele, what is this here? This is the recessive allele is there. What would be there here? Hemophilia is caused. At hemizygous condition, hemizygous recessive condition in male individual, what would be called? Hemophilia is caused. Now you can see female XX chromosome. Here these are the XX chromosomes are there. Now you can notice what is the genes here. Now hemophilia gene. Two are dominant genes. The homozygous dominant condition. What is this? Normal female. What is the female? These two genes are dominant condition. Normal condition is there. Now you can see XX chromosome. One is dominant, one is normal. The recessive it is. So it is heterozygous dominant. Homozygous dominant, normal only. Heterozygous dominant, this is also normal female. But now, what is the case here? Two X chromosomes, both are recessive. Here, this is the two X chromosome. On both X chromosomes, what will be there? Recessive configuration is there. Then how it will be called here? These are hemophilia female. How these are called? Hemophilia females here. These are the hemophilia female. So here on both X chromosome, two dominant genes are there, normal. Both X chromosome on dominant and recessive, heterozygous dominant, normal. But what is the case here? Hemophilia female, recessive condition hemophilia female. And these genotypes for female, these genotypes for male. Now one example we will do this here. What is the inheritance pattern here? If a marriage is held between one, what is this here? Normal woman and hemophilia man, how the inheritance pattern is there? With one example, let us see. If a marriage is held between these cases, example of hemophilia inheritance. Example for this hemophilia inheritance, if a marriage is held between, hemophilia man and normal woman, find the result in F1 and F2 generation. Find the result in F1 and F2 generation. Find the result in F1 and F2 generation. What are the parents here? According to this one, parents. What are the parents? Here, hemophilia man. Male is a hemophilia. Male parent. Two parents are there. Male into female. A cross is done between male and female. What is the male nature? Hemophilia man. This parent's nature is the hemophilia parent. Hemophilia man. But what is the female? Normal woman. Normal female. Normal mother. Hemophilia father. Man means here hemophilia father. Father is here. Hemophilia. Mother is a normal. Hemophilia father means what is the genotype? What is the chromosomes I give? What chromosomes here? Chromosome arrangement. Man means X chromosome, Y chromosome. On X chromosome, what kind of gene? Recessive gene is there. XHY. But normal mother. What is a normal mother here? Normal mother, HH. Or otherwise, H small h. Heterozygous. Simply normal is there. Anyone you take here. Now you have to see this is a normal mother, pure normal mother. Suppose if you are confused, pure normal mother. That means homozygous normal mother. Now, what is the probability to get sperms? Two kind of sperms. What is the 50% sperms? What they have? XH chromosome. Here X chromosome bearing the H allele, hemophilia allele. This sperms Y chromosome, normal Y chromosome. Here this is mother ova is there. What is this here? Normal X chromosome. no hemophilia gene or normal configuration gene. Now here there is a choice to have a cross. This sperm can fuse with this one. Then what is the choice here? There is a, you can see this one fused. What will come? XX, HH will come. The all females are, these are females normal. And next male. What is the male configuration? Normal male. What is the progeny? Once you see this from beginning. If a marriage is held between, what is the case here? This is the case. Father, who is the father? Who is she? Mother. She is a mother. Father into mother. Now you see. Here from this, how many types of sperms can be produced here? XX. It is a sperm. And this is a sperm, Y chromosome sperm from father. And mother, it is homozygous condition. Only one type of ova will come. Now this sperm can fuse with this ova. What configuration? Like this. XX means female. Who is she? Daughter. She is a female is born daughter. But daughter is normal, heterozygous condition. And now you can see what would be born here? XY. What is normal? If a marriage is held between this here, hemophilia father, normal mother. What is the F1 generation? All are normal. There is no problem. Again, a cross is made between F1. This is the F1. Cross is made between F1. So what is the result will come? There is two types of ovas. What is the ova? XX, 50% normal X chromosome, 50% here hemophilia X chromosome. And now sperms, what is the possibility? X chromosome here normal, Y chromosome is also normal. Now you can make a cross. What will come? XX, HH. What is this here? Normal female. Now you can see this here. What is this here? Here this is a carrier female. Carrier female. Over this. Females are over. Next males. What is the male here? This is a male. What is the male? Normal male. The last one. What is the last one here? This is a hemophilia male. That means what is the hemophilia? This grandson is impacted. Here a cross is made between hemophilia father and normal mother. Then F1 generation not impacted. Where impacted? The F2 generation is impacted. Grandson is impacted. Grandson is impacted. This is here a cross is made between father and mother who are impacted. F1 generation not impacted. Grandson is impacted. That means father genes, they are passed to grandson through carrier daughter. Such type of inheritance of it will be called here criss-cross inheritance. This inheritance, how this is called criss-cross inheritance. The passing of father gene into grandson through the carrier daughter, that can be called criss-cross inheritance. Like this, a marriage held between here hemophilia father, normal mother, no impact in F1, but F2 generation, grandson is impacted. This is what is here, hemophilia pattern of inheritance. Thank you, one and all. But up to this hemophilia information, you will be asked the questions on Saturday. Then any doubts are there, you can send a message and seek clarification. Thank you, one and all. And what the concepts here? Color blindness, sickle cell anemia, next one thalassemia, and another one is the phenylketonuria. It will be there. After one more thing, pedigree analysis. Just here, next two classes, I can finish these principles and inheritance of variation. Later, let us see this year, another chapter, molecular basis of inheritance. Thank you, one and all.